Well, welcome everyone to the education portion of the Caring for Carcinoid Foundation's Awareness Weekend. I'm Nancy Lindholm. At 29, almost 10 years ago, I was diagnosed, like many of you here today, with advanced metastatic carcinoid cancer with carcinoid syndrome. So in 2005, I started the Caring for Carcinoid Foundation with the mission to discover a cure for carcinoid and the vision to support all carcinoid and related neuroendocrine tumor patients, their caregivers, and their family members. To achieve our goals, 100% of all individual donations go to scientific research into carcinoid and related neuroendocrine tumors. I believe that we carcinoid and related neuroendocrine tumor patients deserve a future. I believe that each person here in this room today is a hero. From each patient surviving cancer with grace and activism, sitting for long hours in the hard chairs of a hospital waiting room, I've been there with all of you, hoping that you'll be called next, but also at the same time hoping that you will not. From each patient supporter who quietly holds the hand of your loved one during stress and sickness, and also in those moments of sheer joy. From each scientist, physician, or volunteer who works to ensure a better life for each patient that he or she sees. I believe each of you is a hero and deserves a round of applause. I believe that it is up to us, the patients, to spur our physicians, family members, and friends on to support us in the search for a future for all of us, the search for a cure. Let us act and fight together as we move forward to fund cutting-edge research and achieve a cure. Give the gift of a future to your loved ones and to yourselves. You deserve it. Together we will stay strong, stay active, united, and commit to our mission. I'd like to introduce now our wonderful Executive Director, Lauren Erb, to welcome you here this morning and also deliver the to thank the many supporters that made this possible. Thank you to all of you for helping the CFCF achieve its mission of a cure. And now I need a cup of coffee. Thanks, Lauren.
Thank you, Nancy. I'm so excited this morning to see both so many new and so many familiar faces. I look forward to meeting you all over the course of today, and I also would like to welcome you all here to Boston. And I think a really key component of today's day is helping everyone in attendance to help forge those relationships that then you can then take home with you and grow your supportive network. So I'd like to kind of get to know each other a little bit here today. So I'd like to start off by having everybody raise their hand. And then I'd like you to put your hand down if you're from Boston. And then I'd like you to put your hand down if you're from Massachusetts. And now we'll put our hands down if we're from New England. And now we'll put our hands down if we're corporate sponsors. And now I'd like Ruth Gertis and her daughter Jessica to put down their hands. Now put your hands down if you're from farther than the eastern seaboard. Uh-oh. Put your hands down if you're from east of the Mississippi. Put your hands back up if you just put them down. I think we have our winners. And Frank, is that right? Oh, I'm sorry. What was your name? Dave. Dave, where are you joining us from here today? From Pennsylvania. And how about over here? Where are you guys joining us from here today? Ohio. Wonderful. And so I have 2 copies of Carrie's book that I wanted to give to the people that traveled the farthest to be with us here today. Thank you for coming. I have another copy. It's just not in this room. And so, as I mentioned, I do really encourage you all to enjoy each other's companies and to support each other throughout the course of the day. I'd like to thank our co-sponsors, without whose generous support this day would not have been possible. The Network for Patients and Families at the Massachusetts General Hospital Cancer Center. In particular, Sally Hooper and Paula Gauthier, and not only for their support this weekend, but for the support they offer to patients and their families every single day. I'd also like to thank our corporate sponsors, Pfizer, who provided us with a very generous educational gift that is allowing us to produce all the durable materials here today. I'd also like to thank Molecular Insight Pharmaceuticals and also Endo Pharmaceuticals for their sponsorship of today's conference. As you all notice, we are filming all of today's presentations and speakers, so please let me know or let a CFCF representative know if there are individuals that you think would benefit, and we'd be so happy to send them materials from the day's presentations. And so I'd just like to thank you all for coming. Please make sure that you get your questions answered. We have CFCF representatives walking around the auditorium who will be happy to help you. It's so wonderful that everyone could make it out here this morning. Please enjoy the conference, enjoy each other's company, and don't forget, it's about you, the patients, the families, the friends, and the caregivers. And now I'd like to introduce our very first speaker, Dr. Jeffrey Clark from the Massachusetts General Hospital Cancer Center.
This is the last one, yeah.
This is your new one. Oh, yeah, it says
I think I have, can you hear me? No problem. Okay. Thanks very much. I would, and thanks to all of you for braving the monsoon season to make it here. Dr. Ryan was supposed to be here, and he apologizes that he couldn't be here, but he had a commitment that he really couldn't get out of. So I'm substituting for Dr. Ryan. But I've taken care of patients with carcinoid tumors also for over 20 years, so I should be able to answer at least some questions. What I'm going to talk to you about are clinical trials, and I'm going to talk about them in general and then some specifics about the clinical trials we're doing, although that will primarily be covered by Dr. Chan this morning and then Dr. Kulke in terms of more basic research this afternoon. So with that, and the final thing I would do is just echo what Nancy said, and that it's really all of you who are the true heroes of clinical research, because it couldn't be done without you, and you really are essential in the process. So with that, I'll begin. So, probably that. I hope I'll begin. There are a variety of types of clinical trials, clearly, that one could think about participating in. There are prevention trials for patients at high risk for developing a condition. There are treatment trials, which we'll focus on for most of the talk. There are tumor or normal tissue studies, and as I mentioned, Dr. Kulke will discuss that this afternoon. There's early detection or screening, and there's a lot of work going on for us to be able to do that, because one of the keys to curing cancer is to be able to detect it earlier. And it's a very difficult problem, but a lot of research is ongoing. One could improve diagnostics, and then quality of life and supportive care are also critical issues. But we're going to primarily focus on treatment trials. So they really are trying to address the question, what new treatments can help people with a particular disease? And what is the most effective treatment for people with that disease? And the first is really in the realm of the phase 1 and 2 trials, and we'll define those in a little bit. And the latter is in the realm of the phase 3 trials. So phase 1 trials are the first introduction of either a new drug or a new regimen into patients. And one's really asking the questions, how does the agent affect the human body, or how does the regimen affect the human body? What dosage is safe to give? And what is the best dose for us to take into trials where we're going to try to look at efficacy of the agent? In phase 2 trials, what we're trying to answer is, does the agent or intervention. It doesn't have to be an agent. It could be surgery. It could be a new device. But in general, for the most part, the trials in neuroendocrine tumors are new agents. Does the agent or intervention have sufficient effect on a specific disease to warrant further study? And the majority of trials for neuroendocrine tumors or carcinoid cancers are phase 2 trials. And then finally are the phase 3 trials. And in phase 3 trials, one is trying to address the question, is the new agent or intervention better than the standard that exists? Phase 3 trials require a large number of patients to answer questions, and therefore are particularly difficult to give for diseases that are as uncommon as neuroendocrine tumors. Phase 3 trials are randomized. And why do we need randomization? It's essential for us to answer the question. I'll get to that in the next slide. But basically, participants are equally divided into how many of our arms are on the study. One will either receive the standard treatment or whichever of the investigational arms one is assigned to. And the reason it's essential is that one has to try to make all patients on all arms of the study as alike as possible. And the only way one can do that is through randomization. And that's particularly true in diseases such as neuroendocrine or carcinoid tumors, which have such a variable clinical course. And it really does provide the best way to prove the effectiveness of a new agent or intervention. Now, when we do clinical trials, there's a clinical trial protocol that's written, and that's what we really have to abide by in doing the trial. It has strict guidelines, defines the purpose of the study, how many people will participate, who's eligible, how the study will be carried out, and what information will be gathered, as well as what the critical endpoints of the study are. And so some of the benefits of participation in a study is that if the best standard treatment does exist, one is going to receive at a minimum that. There are often settings in which there are no standard therapies, and we're looking at something that's totally investigational. But clearly that's appropriate if there's no standard therapy that one could go on. If the new treatment or intervention is proven to work, then you might be among the first to benefit. And the biggest reason for participating in clinical trials is the last one, is that you really have a chance to help others and improve patient care. And that's really the primary benefit of clinical research. It is the only way we define new therapies. There are some risks to participation in clinical trial. The new treatment or intervention clearly might not be better or even as good as standard care. And then even if the new treatment is beneficial overall, it may not work for every patient. Now, there are ways in which patients' rights are protected on clinical trials. There is the IRB and informed consent. All of our trials go through scientific review. And there are data and safety monitoring boards, especially for all phase 3 trials, that ensure that as the trial is ongoing, there aren't untoward events on one arm or the other that should make one rethink the trial. Now, as I mentioned, scientific review is required for all NCI-designated cancer centers, and certainly for the Dana-Farber Harvard Cancer Center, we have scientific review for all of our trials. Institutional review boards exist at all institutions where federally funded research is ongoing, and they're subject to FDA regulation. The data and safety monitoring boards I mentioned, they ensure that risks are minimized, ensure data integrity, and stop the trial if safety issues arise. They're required for all phase 3 trials. They exist for a subset of randomized phase 2 studies. And then why do so few patients participate in clinical trials? If you look across all cancers in the United States, the percentage of individuals with cancer who participate in clinical trials is in the range of about 3%. And why is that when we desperately need new answers? So some of the reasons are here. Individuals don't know about clinical trials. They may not have access to them. They may be afraid or suspicious of the research. They may not be able to afford to participate. They might not be able to have funds to reach the center where the trial's ongoing. And then in some cases, they may not wish to go against their healthcare provider's wishes. Hopefully that's a relative rarity. There are special issues that apply to neuroendocrine cancers in terms of clinical trials. As I mentioned, it's relatively rare. And so to do phase 3 trials in neuroendocrine cancers is extraordinarily more difficult than, say, in breast cancer, where there are so many more patients who can participate. This tends to be a very variable group of diseases biologically. And so it's difficult to compare results from different phase 2 trials. That you see a phase 2 trial that has, some high response rate and one that has a relatively low response rate, but the low response rate one might actually be a better agent. It's just that the individuals who participate in that study had worse prognosis. It often is a relatively indolent disease, so it takes a very long time for one to get answers. In metastatic pancreatic adenocarcinoma, which has a very guarded prognosis and relatively short survival, one is going to get an answer fairly quickly as to whether something is working or not. For these types of cancers, it may take much longer. And then finally, there's the limited activity of most, certainly standard chemotherapy agents against these diseases. And that has, although there's been lots of research trying to find new active agents, that's limited the ability to move ahead with combinations. But clearly, research is necessary for progress against these cancers. And that research really is going to come from increased knowledge about the biology of these cancers to help us develop new therapies. And this is just a schematic of how this can happen. Is the basic research leads to targets. We can identify lead compounds, prove that they're effective preclinically, refine those compounds, go into phase 1 and 2 trials. And if we demonstrate that they have activity, then one can go on to phase 3 trials to try and establish efficacy. But it's not a one-way bridge. Things that we learn from clinical trials are fed back then to basic research in order to try to come up with new answers. And where we're headed, for the most part, in cancers broadly, is for targeted biological therapies that target specific molecular or cellular changes in tumor tissues for cancer therapy. And the same is true for neuroendocrine cancers. As part of this, we have to develop biomarkers, which are biological markers of clinical benefit. And they really help convert clinical drug evaluation from being empiric, when the only way we know whether it works is the final result of whether patients respond or live longer, to evidence-based, where we can tell whether we're hitting the target or not, whether the tumor is responding on the molecular level. And biomarkers can be used for diagnosing and patient selection with the ultimate promise of individualized therapy. They can help us establish efficacy by predicting or monitoring clinical response to an intervention. And they can help us with safety by predicting and monitoring potential toxicity to an intervention. So there are a wide variety of approaches targeting various aspects of the biology of neuroendocrine cancers that are continuing to be explored, utilizing knowledge that's being learned as we speak about the biology and from basic and clinical research. Now, I can address the trials that we have at the Dana-Farber Harvard Cancer Center, which includes the Beth Israel Deaconess Brigham and Women's Children's Hospital, the Dana-Farber Cancer Institute, and Mass General. And so we have some open therapeutic trials, a couple of trials that Dr. Chan actually will address subsequently. And then there are non-therapeutic trials that are very critical for us to address some of the basic biology of these tumors, and Dr. Kolke is going to talk about those this afternoon. And then there are some trials that are pending, and I'm not sure whether Dr. Chan will talk about these subsequently, but they will be open in the near future. And this just illustrates one of the patients who went on a couple of our trials. It really illustrates a lot of the issues related to clinical research in neuroendocrine tumors. So this was a patient of mine, she's now 54, who presented with essentially a liver replaced with metastatic tumor. And she went on one of our trials that actually proved to be most active, temozolomide and bevacizumab, and she didn't respond. She actually progressed through that. So she then went on a trial that actually was relatively ineffective. In fact, she was the only responder on that trial of pemetrexed. But this is her CAT scan. It continues to shrink 2 years later. And so that she's really had a remarkable response on this trial. It's, as I said, it's not something we would think of taking forward because when one, yes?
Oh, Dr. Clark, I'm sorry to interrupt. Would you just show us on the previous slide where the tumor was?
Sure, sorry
Areas
about
of tumor
that.
in comparison
So this
to
is the tumor
next one.
in the
Those dark spots.
those dark spots, and the normal liver is the lighter tissue. This is the spine and back. This is the front of the abdomen.
It's hard to imagine that someone could live with that much tumor.
It's true, but you can, compress a lot of, as long as there is, sufficient normal liver tissue left, you can compress a lot of it and still live. I mean, her liver tests, function tests were actually fairly just minimally elevated at that time. And then here, this is all normal liver, and these are the same two lesions that were there before, but clearly significantly smaller. We aren't clearly the only place that does clinical trials in neuroendocrine tumors, and I thought I went through the NCI clinical trials website and listed the types of trials that are ongoing right now across the United States. So a lot of the trials are looking at novel somatostatin analogs or the equivalent, plus minus interferon or other agents. And many of the phase 3 trials are in this realm. There are a lot of things targeting angiogenesis,